Hello, my name is Mantasha Ahmed, and today we are going to discuss about how the impulses are transmitted through electrical impulse. Through electrical impulses are transmitted through a synaptic cleft, and how ionic channels and synaptic transmission takes place. Here we know that the basic knowledge that between the two neurons which are not touching, the impulse travel from one position to the second position. As you can see here, the impulse will travel in the synaptic clefts without touching, no touching. Okay, now all this of the mechanism is carried out by impulses, ionic channels. The sodium channel is tightly sealed by one molecule, like there are a molecule of sodium ions. If we take a neuron like this and a effector cell, like there is some cell or an effector organ cell, so it is in contact with that, and how impulse it is travel to it is through a neurotransmitter or some electrical impulse. How it is traveled, we know all about this, we have studied in physiology. Now if we take a closer look, this is a presynaptic neuron, and these are small vesicles of neurotransmitter. Any neurotransmitter, if it is it can be acetylcholine, it can be norepinephrine. These are this is postsynaptic neuron, and the receptors, the green are the receptors on which the neurotransmitter will get attached. Now there is a presence of calcium ions. Calcium ions are present outside of the cell, and when there is some impulse, okay, when there are some impulse traveling, so what happens through Hodgkin and Huxley have told it through exponential relationship between initial change and the membrane potential. Okay, how the peak value of this calcium ion, on the releasing of impulse, the calcium ions will be released. As you can see, how vesicles get transmitted and the neurotransmitter get transmitted in the synaptical cleft. Now this neurotransmitter get attached to the receptor cells, and the work or the transmission is then traveled to the next post neuron and then processed, and which presence in the um, sodium ion release. So we know that sodium how sodium travels the impulses. Now sodium and potassium channels are hypothetical ions, like if we determine the activation and inactivation energies. As you can see a model of here, I am telling you how the synaptic transmission will take place. What we do in this, the postsynaptic membrane of the myoneural junction has been found to contain a high concentration of choline esterase. As you can see in yellow here, it is acetylcholine esterase, an enzyme that catalyzes the hydrolysis of acetylcholine. It has been shown that the receptor substance is a special hydrophobic protein. Now this protein was isolated from the membranes of the nerve ending. It has high affinity for acetylcholine, as you can see here, and other cholinergic substances. D. Robertis was a scientist that proposed a model of postsynaptic membrane. According to D. Robertis, this membrane has discrete receptor regions. The protein receptor, which has hyper hydrophobic surfaces, as you can see here, passes through the lipid layers of the membrane and create channels in it. The wall of the channels are formed by four parallel protein molecules. Acetylcholine esterase is present as an individual molecule. Acetylcholine then attaches itself to the active side of the protein side, and to of its external surface. As a result of this binding, conformational and translational occurs in the membrane, leading to the changes of the ionic permeability. The role of acetylcholine esterase is nothing but mainly consists of hydrolyzing acetylcholine. We know this. If acetylcholine was not hydrolyzed, then we can say We know that through data we can calculate what will actually happen in the postsynaptic and how the impulses are taken. If the number of impulses in which x proportion, as you can see, p is px proportion quanta of ACH are released, then the expression is pxm upon x to the x e to the power minus m by Poisson's law. This is what Poisson's law, where x can be 0, 1, 2, and a number of acetylcholine proportions. M is the average number of proportion released by single impulse in the system study. By calculation, M is equals to the average response amplitude and average upon average amplitude of a spontaneous potential, which is 0.0933 millivolt upon 0.04 millivolt, which comes out to be 0.33. If we coincide the calculated and observed values of this px, like proportions of contact, has been found to be excellent, in particular according to the Poisson's law, which is p naught equals to n naught upon n equals to e to the power m, where n naught is the number of null responses equal to the 18 in the series of consideration, and is the total number of impulses equal to 198. If we calculate this, then it comes out to be 2.4 instead of experimental value which is 2.33. So it's very very close. The quantization of acetylcholine released compels us to believe that a neurosecretion is cooperative molecule process that may be treated on the basis of electronic conformational interaction. The same ideas may prove useful for understanding of the nature of generation of nerve impulse. So